Hello, everybody. Again, welcome to our webinar, Transforming Proteomics with Single-Cell PICO, F.SIGHT, and I.DOT Technologies. Thank you so much for being here today. My name is Holger Armbruster, and I'm the Head of Product Management here at DISPENDIX. Our registrants represent a truly global audience spanning at least 4 continents with particularly strong representations from Europe, the United States, and Asia. And I'm so proud to be moderating this webinar today. On today's agenda, we will hear from an exciting panel of experts in single-cell proteomics. Please feel free to use the Q&A feature through our program. We will address your questions at the end. So Fernando, it's your turn now.
Hello, everybody. Let's start with a general overview of the I.DOT. So what is the I.DOT? The I.DOT is a cutting-edge, non-contact liquid dispenser designed for highly multiplexed, low-volume reagent handling. It dispenses volumes from 2.55 nanoliters up to 30 microliters, offering flexibility for both miniaturized and standard low-volume applications. Compatible with any SBS format plate and even arrays, it adapts easily to various experimental setups. It supports a wide range of liquid types, including challenging reagents like methanol and viscous solutions up to 65% glycerol. In essence, the I.DOT combines speed, precision, and versatility, helping labs boost throughput while minimizing waste and manual effort. Let me tell you some more details while we watch this video. The I.DOT is automation-friendly, non-contact dispenser designed to transfer nanoliter and microliter scale volumes to 96, 384, and 1536 well plates. 96 well source plates can dispense any liquids with viscosities from methanol to 65% glycerol, as said before. Here we see an example of that. The dispense head modulates air pressure, which can be precisely adjusted to generate droplets from 3 to 50 nanoliters at a nanoliter resolution of 0.1. There we see the pressure system. Each channel generates 100 droplets per second into the target well. That makes it a very fast system. A 96 well plate, for example, can be filled with 100 nanoliters per well in only 7 seconds. To save on reagents, the I.DOT has a residual dead volume of only 1 microliter. The dispensed droplets are counted by an optical system, here in the video, providing volume verification for quality control. Along with the I.DOT, today we will also hear about the F.SIGHT. This is one of the many single-cell dispensers that CYTENA has developed over the years, each with some different features that tailor them for specific applications. There's the F.SIGHT Omics for single-cell omics applications, the B.SIGHT for microbiological applications, and the UP.SIGHT and other dispensers of the UP.SIGHT family for stable cell line development. These are all standalone benchtop size instruments. There's also the C-Station, an automated workstation composed of many instruments and software that we will not cover today. Let's focus on the single-cell dispensers like the F.SIGHT. Our instruments dispense single cells on demand using a microfluidics system embedded on a disposable cartridge that generates droplets with single cells that are deposited inside the wells of culture or PCR plates. Once the cartridge is loaded in the instrument and dispensing starts, the nozzle of the cartridge is imaged in real time to select the single cells that will be dispensed. The dispensing process is very fast. For example, it takes 2 minutes for a 96 well plate and only 8 minutes to fill a full 384 well plate. The imaging data is stored to document information for each well to provide proof of single-cell dispensing. I will provide more details and examples later, but let's now hear from Christoph how Actome integrated these 2 instruments to their single-cell PICO assay workflow.
Thank you so much, Fernando. And now, with no further ado, let's welcome our distinguished speakers to the stage. And the first speaker is Christoph Niemöller, who will be presenting Unleashing Single-Cell Proteomics, Single-Cell PICO Revolutionizes Absolute Quantification. Christoph earned his PhD from the University of Freiburg, where his research focused on developing innovative single-cell genomics methods for epigenomic and genomic analysis. Following his PhD, he worked as a scientist at CYTENA, contributing to the development of single-cell applications using precision dispensing technologies. He's currently part of the R&D team at Actome, where he leads the advancements of single-cell PICO assay for true absolute quantification analysis at single-cell levels. So Christoph, take it away. The stage is yours.
Thank you so much, Holger, for the kind introduction. I'm really happy to be here today to give this webinar about our protein interaction coupling technology, short PICO, and our recent development of proof of concept workflow, which we have developed to apply PICO in order to precisely measure proteins within single cells, also using the F.SIGHT and I.DOT technologies. So what is protein interaction coupling, short PICO? Actually, it is a quite novel, powerful technology which combines the advantages of an immunoassay with a digital PCR readout. So what do? We actually employ highly specific antibodies which bound to our target protein, and these antibodies are specifically labeled with DNA oligonucleotides. There is it. So these antibodies are labeled with DNA oligonucleotides in order to facilitate the detection of our protein target of interest using digital PCR. So how does it work in detail? So in our complex samples, we have a target protein, our protein of interest. And this protein of interest is targeted by 2 antibodies each in our assay. That's an important point to highlight here. 2 antibodies bind simultaneously to the target protein. So the first step is mixing of the 2 DNA labeled antibodies with the sample in order to facilitate binding. Then in a second step, this binding reaction is heavily diluted and compartmentalized. And ideally, the compartments where those target proteins and antibodies are being partitioned in usually should only contain 1 or less target protein or target DNA oligonucleotide per partition. And within those partitions, we perform a PCR amplification. This is a digital PCR amplification. And this results in single positive partitions once there was only 1 oligonucleotide or 1 antibody present within the compartment, here in yellow and blue, or in double positive partitions. And assuming that the distribution of the antibodies within the partitions is just random, we would assume that the double positive partitions, the number of double positive partitions, is a result of a pure random distribution, a Poisson statistic distribution. However, since some of the antibodies are always engaged in binding our target protein, we see a divergence from the Poisson statistics. And this is how we actually calculate our target protein abundance within the sample. So using a quite complex bioinformatic tool, which is also available online, we are able to analyze our data precisely from the dPCR output to precisely quantify the target protein abundance. So what can actually be measured with PICO? So the first scenario is the simplest one. You have a target protein which are bound by 2 antibodies as described. In the second scenario, we can also measure post-translational modifications by employing 1 antibody which is targeted against the protein and 1 antibody which is bound or which binds to the post-translational modification. In the third case, we can also measure protein interactions by using 2 antibodies which are specific for each partner, each interacting partner of a protein complex. And there is actually also a fourth scenario, and one which I want to focus on today, which we call a triangular assay. In this kind of assay, we can measure the protein abundance and the post-translational modification of the same protein at the same time. So simultaneous measurement of protein abundance and post-translational modifications by employing 2 antibodies which are targeted against the protein, and 1 antibody which is targeted against, in this case, the phosphorylation of 4-BP1. And this results in 3 distinct readouts per experiment, 2 readouts for the phosphorylation and 1 readout for the protein signal. So what are actually the advantages of PICO over traditional methods as Western blot and ELISA? First of all, we are able to offer true absolute quantification, so there's no external standard needed. We can quantify, as described, complex target, protein-protein interactions, and post-translational modifications. It's a multiplex analysis, which means we are not only able to measure one signal, but several signals simultaneously. It's compatible with a high throughput assay, so you can use up to 96 well-deep PCR plates. It offers high sensitivity, requires only little material input of 1 microliter, high specificity, which is also really important to note. We are using 2 antibodies at a time, which have to bind simultaneously to the target. And if there's unspecific binding, we do not see any signal. And we offer online data analysis for flexible assay design and analysis. Of course, we offer also PICO kits, which basically cover the whole workflow. So if you're interested in trying out PICO, please get in touch. First of all, the PICO glue antibody labeling kit, which is compatible with the PICO glue labels. So this is a highly efficient method we have developed basically for labeling our antibodies, which usually yields up to 100% antibody labeling efficiency. And we also offer the PICO amplification core kit and the PICO probes, which are then used for sample preparation and for dPCR amplification, which is then, of course, followed by bioinformatic analysis using our online tool. So let's jump into the applications. Measuring post-translational modifications as described with our triangular assay. In this case, we have been measuring 4-BP1 and its phosphorylation, and we applied this in a bulk assay, so this is regular PICO, to BT474 cells, which were either treated with DMSO, so the mock-treated cells, or with dactolizib. And dactolizib is a quite potent PI3K mTOR inhibitor, which should diminish the 4-BP1 phosphorylation signal. And indeed, what we could see is that in comparison to the mock-treated cells, we see a strong reduction and highly significant reduction of the phosphorylation signal in the treated cells. So just as a short wrap-up, PICO offers true absolute quantification. It's highly sensitive. It offers high specificity. And this is why we actually came up with the idea, why not developing a proof of concept workflow to apply all those advantages to the single cell level in order to undercover cellular heterogeneity on the protein and single cell level, in order to enable complex pathway studies on a single cell level. And for this, we used the F.SIGHT and I.DOT technology. So how does this workflow actually look like? In the first step, we dispensed only 12 nanoliters of our antibody mix, which also contains lysis components, into a 384 well plate using the I.DOT. Everything here is covered with vapor lock to ensure that there is no evaporation of the droplets. Then in a second step, we use the F.SIGHT to precisely deliver single cells to those tiny droplets. Then we do an overnight incubation step where binding of the antibodies takes place. And then the rest is followed by a quite regular standard PICO assay. So dPCR analysis and then bioinformatic analysis. One very crucial step within this workflow is to control the efficiency of single cell isolation. As you can imagine, we are trying to target a droplet which is only the size of 12 nanoliters under a vapor lock encapsulation barrier. So this is not easy to do. but it works using the F.SIGHT. So the F.SIGHT is able to precisely deliver tiny droplets which contain the single cells of interest, as you can see in the picture in the middle, precisely to the center of the well. And of course, this is not 100% efficient, but it's still highly efficient for the given volumes. And we control this delivery of the single cell containing droplet to the antibody mix by using a fluorescent readout. So we check if there's increased fluorescence within the droplet when there was a single cell delivered to the antibody mix containing droplet. So, and these are actually the results we were able to measure for the single cell application. So we again applied our triangular assay, a single cell PICO, using 2 anti-4BP1 and 1 anti-phospho-4BP1 antibody to 2 times 40 single U937 cells, and again treated them with dactylizib or left them untreated. And I just want to remind you that in this case, each of the dots represented in the graph represent a single cell measurement. And on the left-hand side in A, you see the raw or the corrected Kuplex counts, protein counts. And on the graph on the right-hand side in B, you see the absolute quantitative amount of targets per cell. And what we could see is not only that there is already a significant level of heterogeneity amongst the cells if they are untreated, but there's also a significant reduction of the phospho-4BP1 levels induced by dactylizib, and there's also striking heterogeneity in response to dactylizib. And with this, I would like to summarize. So PICO enables precision proteomics in a kit. We are actually the only method which is able to offer true absolute quantification of proteins. It's highly sensitive, it's highly specific, and we applied those advantages of PICO to the single cell level, establishing single cell PICO to precisely quantify 4BP1 and 4BP1 phosphorylation levels in single cells. We saw a significant level of heterogeneity amongst the cells and upon treatment, and this was, last but not least, enabled by the F.SIGHT and I.DOT technology. So where can you actually read more about PICO? I just want to highlight that we just recently published a major paper where all the details of the PICO technology is presented, available online at Scientific Reports. We already published a few application notes. Everything can be found here by scanning this QR code. And these are our academic partners. And with this, I would like to thank you for your attention. And I would like to close. And please get in touch if you're interested to try out PICO yourself. We are also offering a trial kit. So please get in touch if you're interested. And thank you.
Okay. Thank you so much for that insightful presentation, Christoph. So our next panelist is Fernando Aprile García, who will be presenting integration of I.DOT and F.SIGHT into high-throughput workflows. Fernando Aprile García is a seasoned expert in cellular models and cell line development, driven by a passion for advancing modern therapeutic innovations. His journey with CYTENA began as a senior application scientist, and he now leads the applications team, where he combines technical expertise with leadership to optimize workflows in cell line development. So, Fernando, the floor is yours.
Thank you, Holger, for the kind introduction, and thank you, Christoph, for showing us such a powerful technology, the PICO assay, and its application at the single-cell level. As you might have noticed by now, the common themes of this webinar are single cells, assay miniaturization, and high throughput. It was very interesting to see how the F.SIGHT and the I.DOT were used for sample preparation, enabling a high throughput, low volume single cell assay. I will continue with this theme. I would like to showcase some other examples of how the F.SIGHT and the I.DOT can be integrated in high throughput workflows for more precision, reproducibility, and usually at a lower cost per cell or well. So first of all, for our proteomics audience and in connection to Christoph's talk, I wanted to quickly mention this publication from 2023, where the I.DOT was also integrated in a single-cell proteomics workflow. In this work, a new DIA method was developed, including low input samples and low volume dispensing, where the I.DOT became very useful. But the I.DOT is not only useful for single-cell proteomics, like this example, or like Actome's single-cell PICO assay. Let me mention the target applications where the I.DOT provides a lot of value. Genomics applications are improved by the I.DOT mostly due to precise dispensing of qPCR master mixes and preparation of NGS libraries. Drug discovery speeds up when the I.DOT is integrated in assay development workflows and in experiments with dose response curves. And the I.DOT can also be used in synthetic biology applications for gene synthesis, for example. I want to highlight the NGS workflow in genomics applications because it shows very well how the I.DOT can replace tedious, error-prone, and expensive manual pipetting during library preparation. DNA library preparation for NGS usually starts with a low amount of DNA sample and require very small volumes per well. When high throughput, several 96 or even 384 well PCR plates need to be processed, all the steps that require a reagent addition can be performed with the I.DOT. Fragmentation, adapter ligation, PCR master mix dispensing for PCR amplification, and sample normalization to have the library ready for sequencing. Along this workflow, there are cleanup steps needed to purify, wash, and size select the DNA sample. These steps are classically performed manually by the scientist with a magnetic plate holder and a hand pipette, something that can lead, of course, to interwell variations, time loss, and the considerable use of pipette tips. A solution to this is to include an automated plate washer in the workflow, like the GPure from DISPENDIX. Very briefly, the GPure is an automated plate washer for non-contact liquid removal and dispensing. It can process 96 and 384 PCR plates in under 2 minutes without the use of pipette tips. It's compatible with all NGS protocols out there and magnetic beads that are there in the market. So when preparing libraries for single-cell omics, this workflow can be expanded with the integration of the F.SIGHT at the beginning to dispense the single cells onto the PCR plate. Here we see such an example of a single-cell RNA-seq workflow automation. This work was carried out at the IOB in Basel, Institute of Ophthalmology Basel. There, they developed a user-friendly automation workflow for full-length single-cell RNA sequencing with the FLASH-seq method. For this study, the material source were retinal organoids that after dissociation, the single cells were dispensed with the F.SIGHT onto PCR plates with only 1 microliter of lysis buffer. On these plates, the library preparation for the FLASH-seq method was performed with the I.DOT and GPure, like I showed you before. Libraries were then sequenced, and the typical clustering analysis of gene expression in single cells was performed to characterize the different cell populations on the retinal organoids. The steps of plate prep, cell dispensing, RT-PCR mix dispense, cDNA cleanup and normalization, fragmentation and enrichment PCR were automated and streamlined with the I.DOT and GPure. This led to considerable improvements on the method that are discussed on this application note, and that includes assay miniaturization, meaning less cost per cell, and also assay automation, leading to faster, less hands-on time, higher throughput, and enhanced reproducibility. Okay, so now that I mentioned the F.SIGHT, this gives me the opportunity to move on to the second part of my talk, where I would like to discuss the single-cell dispensers and give a brief overview of the technology and its applications. As mentioned at the very beginning of the webinar, CYTENA's dispensers dispense single cells on demand using a microfluidics system embedded on a disposable cartridge that generates droplets with single cells that are deposited inside the wells of culture or PCR plates. Cells can be selected according to size, roundness, and fluorescence intensity, given that the system is fitted with a laser to illuminate cells that are stained or expressing fluorescent markers. Here you see a real-time video of how single cells are dispensed. You can see how fast it goes. One can actually see what is being sorted. The system provides images of the events pre, during, and after dispensing to document the single cells that get dispensed. We can have a quick look at how the dispensing mechanism works in this video. Between the nozzle and the culture plate, there's a vacuum pump that is always on. And there's a piezo controller in the dispenser that generates droplets one after the other at the nozzle of the cartridge. The content of every droplet is analyzed in real time by this imaging system. If the algorithm doesn't detect any cell or detects two or more cells, the vacuum is turned off. It's always on, and then those droplets are sucked away. Conversely, when only one cell is detected by the detection system, then the vacuum pump is turned off, and that droplet is allowed to land on the well of the plate. This will happen for every well because the plate stage will move one position and the dispensing process continues for each one. As you might have noticed by now, at the core of these instruments lies this disposable dispensing cartridge. This is where cells are loaded, and the only part of the instrument that comes in contact with the cells, eliminating the risk of cross-contamination and making the instrument very easy to clean and maintain. Here we see a video of how easy it is to load the cartridge. The cartridge is loaded onto the dispenser, which has a piezo controller that creates these small droplets I was telling you about, containing the single cells. This leads to a very gentle dispensing process without the need of high pressure, such as those seen in flow cytometers, assuring higher cell viability and the possibility to dispense rare sensitive cells. The cartridge holder, as you see in the video, is magnetic. This makes its handling user-friendly, and the setup time is quite fast. So, as mentioned before, CYTENA has a variety of single-cell dispensers with some different features that tailor them for specific applications. So let's now focus on which applications each dispenser excels at. The F.SIGHT omics is tailored for single-cell omics, such as single-cell proteomics and transcriptomics. This is because of its key feature of automated offset compensation, short AOC. This is a quality control system that adjusts the droplet position by detecting how far from the center of the well the droplets are landing to then adjust the dispensing direction. This ensures precise and reproducible cell deposition at the center of each well, either 96 or 384 well plates, therefore enabling downstream miniaturization of cell lysis buffers, for example, in sub-microliter volumes, even less than 1 microliter, like Christoph just showed in his presentation. The cell will land on the very small surface of that 1 microliter lysis buffer at the very bottom of the conical well of a PCR plate. So with this technology, several laboratories are already publishing studies and methods describing automation and miniaturization of single-cell omics methodologies. Here a couple of those examples. CYTENA has also developed a single-cell dispenser optimized for microbiological applications, the F.SIGHT. F.SIGHT enables bacteria, yeast, and small-sized microorganisms dispensing because it's fitted with the dual-channel high-resolution microscopy system that images the nozzle in bright field and fluorescence. The cartridge is adapted to small-sized particles, and the higher magnification over the nozzle allows detection of small cells like bacteria. The system is compatible with bacterial culture in anaerobic conditions, since it can be easily placed inside the usual anaerobic chambers due to its benchtop size. The B-Sight replaces the tedious, lengthy, and inefficient process of manual plate streaking. This is a game changer for 3 microbiology applications, namely strain development to generate new genetically engineered strains faster and with less resources, microbiome research, since one can isolate single cells from mixed samples to culture and characterize new strains, and single-cell genomics from complex samples without the need to culture and enrich these microorganisms. I invite you to check these papers if you have interest on the B-Sight to get a feeling of how this instrument simplifies these applications and enhances output. So for the last part of my talk, I would like to focus on the F.SIGHT second generation. Unlike the other instruments, the F.SIGHT second gen is 2 instruments in one, actually. Combining single-cell dispensing capabilities and plate imaging capabilities. This makes it ideal for stable cell line development applications due to some specific needs these fields have that I will now discuss. The overarching goal of cell line development is to generate a stable, genetically engineered cell line in order to produce advanced medicines, biotherapeutics, such as monoclonal antibodies, viral vectors for gene therapies, or cell therapies. Classically, this process can take between 6 to 18 months from gene to master cell bank, depending on the level of automation and overall process. On top of this, in order to fully cover the requirements from the regulatory health agencies, the producers of biotherapeutics need to provide proof that the newly developed cell lines were derived from a single cell, the so-called assurance of clonal derivation. In the early days, single cells were placed on culture plates either by the method of manual limiting dilution or flow cytometry. Both are inefficient and cumbersome methods and lead usually to very low recovery of clones. Clonal derivation assurance was documented by imaging the bottom of the plate after plating the cells. We now can do all of this with a single instrument, the F.SIGHT second gen, since it's a single-cell dispenser that also provides image-based evidence of clonal derivation composed of 2 imaging systems. First, imaging at the nozzle level, like we've seen so far. This documents that only 1 cell gets dispensed. And to provide the double assurance, the bottom imaging system of the F.SIGHT offers 2 imaging modalities to document that there is only 1 cell in each well. Either a new imaging method developed by CYTENA called 3D well imaging, where an image stack is acquired directly after dispensing and the cell is captured by any of these images while sinking to the well bottom, or the more traditional well bottom imaging once the cell is located at the bottom of the well. The bottom imaging system of the F.SIGHT is not only useful for clonality assurance on the day of dispensing, one can also easily track growing colonies, no matter if working with suspension or adherent cells, as you can see in these example images. And also the imager can be used for cell count and conference analysis with algorithms that we have developed and an analysis software that will provide these cell count and conference measurements. To close, we carried out a validation study to challenge the instrument and prove what it's capable of in a cell line development environment. This study is available online, and it shows that under optimal conditions, the F.SIGHT achieves a single cell dispensing efficiency higher than 97%, meaning that almost all of the wells of any given plate are filled with single cells. Proves monoclonality so well that it virtually produces no errors, and we can always document that there is a single cell on those wells. And it can result in up to 80% cloning efficiency, meaning percentage of colony outgrowth after dispensing. This is due to what I was telling you before, that the dispensing method is so gentle, and then cells can recover very nicely and show high viability afterwards. So this study illustrates the fact that cell line development campaigns become incredibly efficient and compliant with the regulations when the F.SIGHT is used to generate new cell lines for production of biotherapeutics. So to wrap up this part of the webinar, I hope I gave you a glimpse of the different flavors in which our single cell dispensers come, each being the optimal choice for different applications, and how they can be integrated in different high throughput workflows, sometimes in combination with the I.DOT. That's it from my side. Thank you very much for your attention, and I'm happy to take any questions in the final part of the webinar.
Thank you so much for your insightful presentation, Fernando. And now it's time for the Q&A, and we can jump in and answer any questions that you might have. As an icebreaker, I would start, and that's a question for you, Christoph. So a long time ago, I was also doing signal transduction research in cancer and colorectal cancer, and I was using different technologies. I was using Luminex and Western blotting. And for me, one of the challenges in that work was to get reproducible results. And that depends on different factors. It's on the cells themselves, right? On their mood on that day, on how consistently you cultivate them. But then also it also depends on the technology, right? So I was wondering if you compare the PICO or the single cell PICO technology with more traditional technologies like Luminex or Western blotting, how does this compare from a technology perspective, excluding the part of the variation of the cells themselves?
Yeah, thank you so much for this question. Yeah, actually, PICO is highly reproducible. I mean, if we're talking about bulk assays, then we basically measure the same signal over different people conducting the assay in different labs, if we are using the same dilutions, of course, and the same sample preparation steps and so on and so forth. So this is highly reproducible. It's also shown in the paper I just mentioned in the end of my talk, where we show that different conductors basically yield to the same results. Yeah. For the single cell assay, it's a bit more tricky to answer this question because it's hard to measure reproducibility on the single cell level, because each single cell is actually per se a biological replicate, right? So of course you can measure a larger population and see if the CV, for example, or the median signal is similar, and it is. But for the single cell world, reproducibility and signal comparison amongst single cells is always a bit tricky. Yeah, but the assay itself, the technology itself, offers a high level of reproducibility.
Great, thank you so much, Christoph. Cool. Yeah, I mean, if you do have questions after the webinar, feel free to reach out to us, and we're happy to consult you with any things that popped up during the webinar. And with that, I would like to thank you all again for attending the webinar today. We thoroughly enjoy connecting with the scientific community, and of course, we'd love to hear your feedback. And there will be a short survey and webinar recording included in a follow-up email. So have a great rest of the day, everyone. And please look out for the short survey coming in the next days via email. And with that, have a great rest of the day. Hope to talk to you soon. Bye-bye.